The overall goal of this procedure is to conveniently isolate unnatural amino acids from a Ni-Schiff-Base complex and subsequently Fmoc protect these amino acids.
This method can help answer key questions in the organic chemistry field, such as how to more easily synthesize unnatural amino acids. The main advantage of this technique is that it allows for isolation of amino acids with acid-labile side chain protecting groups from a Ni-Schiff-Base complex.
Individuals new to this method will struggle because there is a limited solubility of the nickel complex in organic solvents. DMF is the most appropriate solvent because it can dissolve the complex and is miscible with water.
We first had the idea for this method when we received insight from our inorganic professor, Dr. Boron, about nickel and metal chelating agents.
To begin this procedure, add 40 milliliters of DMF to a 250 milliliter round bottom flask. Dissolve 1 millimole of the nickel PBB Schiff-Base complex in the DMF with stirring at room temperature. Next, add 60 milliliters of 0.2 molar aqueous EDTA solution at pH 4.5. Using a magnetic stir bar and stir plate, stir the solution overnight. After the reaction is complete, transfer the solution to a 250 milliliter separatory funnel. Add 50 milliliters of DCM. Then, cap the separatory funnel and mix. Drain the organic wash into a waste beaker. Repeat the process of adding DCM, mixing, and removing the organic wash 3 times. After this, collect the remaining aqueous layer in a 250 milliliter round bottom flask.